There's still one speaker missing. I hope he'll show up. But, well, yeah. All right, so welcome, everyone. This session is on mathematical modeling and computation design tools. Design is one of the hallmarks in synthetic biology. It is a lot to do with how you design new systems, how you redesign older systems with newer functions, and to all that, as to design of any electronic component, you need mathematical models and tools. There's a wealth of mathematical models and computational tools developed for systems biology, for example, trying to understand all the connections among different components in complicated networks, whereas systems biology tries to provide some understanding of how these components are connected to each other, what are the emergent properties coming out of them. You still need to use some of these and other tools to the specific purpose of synthetic biology. If you have, for example, circuits, you want to combine them in different ways, you need very specific tools. So this session is specially dedicated to make the transition from some of the tools you use in systems biology and to identify some of the formal tools you would need in synthetic biology. That's, of course, by no means an extensive overview. We have 3 selected talks here, and I hope you would... So the first one is by Philip Wright at the Sheffield University. So we'll introduce more the systems biology type of modeling approaches, making the connection to the synthetic biology. Will be followed by Carolyn Lamb from the Imperial College, formerly now at the Helmholtz Institute in Braunschweig, Germany. She'll focus more on formal design tools for synthetic biology, specific circuits, and then we'll have Alfonso Jaramillo from the École Polytechnique in France, and we'll provide a palette of formal design tools by automated design of genetic circuits. So I hope these 3 talks will give you an interesting overview of how we move from, let's say, the more systems to the specific design tools in synthetic biology. Thank you. So the first one is Philip Wright.
Thank you very, very much for coming this afternoon. I'd like to thank Victor particularly for the invitation to speak this afternoon about some of the ideas we've got about combining systems biology and synthetic biology, and hopefully using some of the tools that we've been developing in our systems biology studies and to see what sort of application they might have in synthetic biology. And so just to remind all of you, I guess most of you should know this, what systems biology is, but systems biology really seeks to quantitatively understand the dynamic relationships between various components within a cell. And by components, I mean you might be trying to understand all of the proteins in a cell and all of the mRNA species in a cell and look at the interactions between them, and then hopefully try and get some sort of understanding when one's looking at these particular species, what's happening to the overall phenotype of the cell itself. And really, the success or the underpinning idea behind systems biology is this iterative relationship between wet experiments, so experiments that biologists and biochemical engineers, etc., are carrying out, and between modelling people. And I think really it's been a field where it's been necessarily built on this iterative relationship between models informing experiments and experiments informing models, and really it's the iteration between this that's been the real success of the field. And throughout the world, there's a large number of organisms, microorganisms I'm talking about now primarily, that have been examined from a systems biology perspective, and really this field is growing. And as you look at these particular organisms, obviously some of the most famous ones are things like E. coli and yeast. And so the practice is certainly being in metabolic engineering and now in the synthetic biology field that these organisms are primarily now, particularly the top couple, I guess, are emerging as particular chassis that are being used for synthetic biology applications. But obviously, as more understanding in some other types of organisms are emerging from systems biology studies, there does exist now the opportunity to formulate chassis based on perhaps some other organisms, because they're going to have maybe useful properties that it may not be so easy without really extremely large-scale modification to actually use as synthetic biology chassis there. And you can see that really it's quite diverse, coming down even into the archaea, and the particular types of organisms that we're very, very interested in terms of the synthetic biology projects that I'm involved in are the cyanobacteria, and there are a number of different examples of those. I've given a couple of unicellular examples here, but there are also sort of filamentous organisms as well, and I'll touch on that a little bit later on throughout as the talk goes a bit further. So synthetic biology, I guess, being at this conference is kind of obvious, but synthetic biology is a very, very much an engineering design kind of mindset. It's very much about the creation of tools to develop products, really, or at least that's the way I view it. And physical sciences and engineering tools of systems biology that created the quantitative field. I think it's really, really ripe to take that information and that knowledge and really take it forward into the sort of products generated focus of synthetic biology. The particular area that I'm involved in synthetic biology primarily is the production of biofuels, and in particular using cyanobacterial chassis for the production of biohydrogen. And of course, hydrogen can be produced in other types of organisms. You can make it in E. coli through fermentative processes, for example. But really, I think it is interesting from our point of view, really, is to try and hopefully take sunlight and develop biofuels such as hydrogen. And of course, one could make liquid fuels like ethanol and butanol and other things as well, I suppose. We've heard quite a bit about those kind of fuels at this conference so far, but our focus really is on conversion of solar light using cyanobacteria as a synthetic biology organism. And this is really part of a larger consortium, and you'll hear Alfonso talk at the end, I'm sure, about the Biomodular H2 Consortium, which is an EU, European Union Framework 6 NEST Pathfinder project, which is really focused on this use of a cyanobacterial chassis to actually produce biological hydrogen. And there's a number of different organisations, Freudian slip perhaps. A number of different organisations involved, and these organisations have particular skills, whether it be experimental in terms of sort of systems biology work, molecular biology work, right through to modelling. And I'm from the University of Sheffield, and a lot of the work that we're doing on this project is characterisation of system-wide behaviour when we've implemented some of the synthetic biology changes within the cyanobacterial chassis. And so the overall aim of the project is sort of to put it into a couple of different sort of rather long bullet points. But the project aims at designing reusable, standardised molecular building blocks to produce a photosynthetic bacterium containing engineered chemical pathways for competitive, clean and sustainable hydrogen production. And really, the other thing that we want to do, so that's one of the main aims. So we want to make this device or this system that's going to make a product, but also what we want to do is we want to, in the second bullet point there, what we really want to do is we really want to also develop some tools so that we don't need to reinvent the wheel each time we want to create a new product. Okay, so we want to make some generic tools, and I'll present a little bit of that today, and Alfonso will present some more at the end, so that when we want to go and produce another bioproduct, say, a liquid fuel or a carotenoid or whatever it might be, we don't need to go and reinvent the wheel. We just pick up this toolbox and move forward to produce whatever particular other bioproduct that we might have of interest. And so it's a fairly complicated business, really, because there's a number of different networks that one needs to work on when trying to produce hydrogen in a cyanobacterium. It does produce it naturally, but it's producing it at such a low level that it's not really commercially useful. And so, one needs to make quite a lot of changes here and there throughout the system to be able to understand, to actually optimise the production. And the problem is that the understanding of the hydrogen production process in cyanobacteria is known, but in terms of how to get deep control of this, it's not so easy to do, and the information really is a little bit scarce. And there's a few different things that we're focusing on in the Biomodular Consortium, and part of this is the efficiency of the energy conversion process. There may be some requirements for various metabolic switches, because one of the particular problems that cyanobacteria have, and the hydrogenases in general often have, whether they be algal or whether they be cyanobacterial, is that they're very, very oxygen sensitive. As it turns out, the cyanobacterial ones are often reversible, whereas the algal ones often are not. But nonetheless, so we need to be able to work with a situation where we might need to design various devices for oxygen scavenging, etc. And what we need to be able to do is we need to be able to understand the impact of these large changes on the organism, so that we can hopefully then, again, iteratively then work towards a more optimised design to produce the biological hydrogen. And unfortunately in biology, a lot of the things that happen are due to various types of interactions that happen in the cell. So changing a whole raft of genes in isolation might have unpredictable results. And so we want to be able to understand what the perturbations that ripple out through the system are when we do when we do a genetic change, for example, and to then hopefully mitigate against these through an iterative design process and hopefully eliminate them. And one of the things, we've done a lot of systems level work in a range of different organisms in my laboratory, and what's become clear to us over the years in working with all of these different types of organisms here, whether they be the hyperthermophilic archaea in production of alcohols, or whether it be production of carotenoids and polyketide synthase type compounds in cyanobacteria, or whether it be enhanced production of ethanol in high gravity fermentations in yeast, is that it's been very, very clear that taking high throughput data such as transcriptomic data and particularly proteomic data, which is mostly what I work on in my laboratory, trying to take that and make sense of it in an engineering sense and then try and use that to optimize a process is not so easy, because it's one thing to make the measurements and sort of say, okay, under a high gravity fermentation process, say in yeast, these particular proteins are upregulated because of stress. Okay, so that's one thing you find out. But then it's another thing to sort of say, well, what do we actually do about that to try and mitigate against that and actually generate more products? Because obviously in the synthetic biology, we're more interested in products, whereas in systems biology, we're just trying to understand what's going on. At least that's sort of my take on it. And I put this slide up because we do a lot of proteomics work, and I'm a chemical engineer by training, and I like to work at the business end of something, so I want to work, as close to the actual process as possible in lots of ways. And I think this comment appeared in Nature just over 3 years ago, and it's something that that I think is very much our lab philosophy. That is, at the end of the day, proteins, not genes, are the business end of biology. So that may be controversial for some because there's obviously a lot of focus on transcriptional microarrays, but I do think that proteins are very, very important. And I'll try and convince you of that, perhaps. And, obviously in the central dogma of molecular biology, the protein process sits here in the middle. And so I'm not saying, and for systems biology studies, we do need to look at all of these different pieces of information, DNA, the actual message, the metabolite, the metabolites in the cell, and obviously the quantitative modelling framework. But where we often start when actually trying to find out what the phenotype's doing is we often start at the proteins, because they're the business end of the cell, and then we might move either up or downstream in the process to then look where some of the other control mechanisms might be. And there's just a few comments I wanted to make here about why one should look at proteomics rather than transcriptomics, or at least start there. I'm not saying one shouldn't do transcript microarrays, we certainly do, but post-translational modifications are an extremely important aspect of the cell. You can't pick these up with transcriptional microarrays. There can be multiple gene products from the same gene, even in the bacteria, so that's another reason why it's important. The half-life and the I guess turnover rate, half-life of mRNA is extremely different to proteins most of the time, and so what you find is that there is, and there's numerous papers that present this in the literature, is that there is often extreme discordance between what you measure at the mRNA level and what you measure at the proteome level. And so one needs to be very, very careful in making some inferences about what is linking the phenotype of the cell to what's measured at the RNA level, because it's certainly often not at all what's happening at the proteomic level of the cell. And we've done numerous studies where we've seen very poor correlation, and so one does need to be very careful. And, proteins last a lot longer in the cell. So, the behaviour that might have been transmitted from the message into the production of that protein will be having an impact on the phenotype for a lot longer than when the mRNA's actually disappeared. Transcriptomics is a very, very useful tool, though. It's an awful lot quicker than proteomics, and so when doing a lot of experiments, perhaps it might be of an advantage. The instrumentation is a lot cheaper. It's still expensive, but it's an awful lot cheaper than a tandem mass spectrometer. The problem with proteomics is that there's a massive dynamic range in the proteins that are in the cell, and so it's very, very difficult to try to identify very much of the proteome. Whereas with mRNA, you can essentially get full coverage of what you want to observe, because you know what the genome sequence is, and you can design probes, and amplification can happen via PCR, and so it's a lot easier to deal with, in fact. And so the problem is that when you generate a lot of data from proteomics, I'm talking about mainly here, but it can also be from transcriptomics, is that manual exploration is often painful. It can take several weeks longer to actually do the analysis of the systems level information than to actually do the experiment itself. One needs to, unless very careful examination is performed, it's hard to see how different upregulated branches of metabolism are connected to each other due to the intricacy of the network. And the routine exploration of how things are perturbed in a network when, say, synthetic biologists are making large numbers of changes is something that we need to rapidly improve, because it's going to be very, very painful to do large numbers of these analyses. And so it's something that we really do need to be able to solve if the field's to move forward rapidly. So when one is making, when one is trying to make measurements of the proteome, there's a number of different approaches that one can use to quantify what's happening in the proteome, and they're listed down here. I won't really go into them too much, except to say that we mostly use, certainly for high throughput experimentation, an isobaric mass tagging approach to be able to identify what's happening in the proteome. And for that, we routinely obtain, I don't know, 400, 500, 600 protein identifications and quantifications within the time frame of a particular experimental set. And certainly with, if you're working with E. coli or various other types of organisms like that, it's routine to get anywhere from 5 to 20% of the protein quantified by the methods that we typically use. You can get an awful lot higher coverage of the proteome, 50% maybe even higher, by spending an awful long time, weeks and weeks of mass spectrometry, to be able to obtain much, much higher coverage. But in routine operation, when doing high throughput quantitation, anywhere from 5 to 20% of coverage is pretty typical. And one of the problems with that you'll observe, of course, is that therefore, because the coverage is so low in comparison to microarrays, where you see more or less everything, is that the data is very, very sparse. And what we've been concentrating over the last 12, 18 months or so is trying to develop some tools which have just come out in bioinformatics. Actually, the bottom one's just come out this month. Is a range of different tools to be able to handle this sparse data and then provide hopefully upgraded information to systems biologists and synthetic biologists so that greater insight into the cellular behaviour can be obtained. These models are based on certainly initially what we've been doing is looking at metabolic network structure. And metabolic network structure has a number of different features. Is that we've been basing this on the enzymatic reactions that take place in the cell at the moment. We haven't been focusing too much at the moment on regulatory networks, but it's something that we're looking at. So we're mostly looking at metabolite reactions. The main features of the network structure is that it has a small world network structure, and what this means is that the average path length is short within the organism, within the network. The original design was based on sociological networks, this so-called 6 degrees of separation approach. And basically what this means is that the path length between one metabolite and another metabolite somewhere in the system is likely to be quite short, because there are a lot of very well-connected hubs in the system through which these metabolites can transfer. So the difficulty of working with a network structure when you're working with proteomic data and trying to determine, say, upregulated pathways or downregulated pathways in a sparse network is this particular observation here. So the red circles are the upregulated proteins. The open circles are proteins which are not differentially expressed. The little blue one over here is a downregulated protein. This is an a priori network that one might have obtained from a genome reconstruction, say in E. coli, like a Palson model, or it might be something that you've obtained from KEGG, for example. But what I want to draw your attention to is all the black dots. These are the things you didn't find. So because you didn't find them, you've got no idea what they did. Did they go up? Did they go down? Did nothing happen? And this is really a typical sort of look at what a proteomic data set will look like when overlaid over a metabolic network, and that's a big problem. So the first tool we developed was a heuristic tool to actually try and construct this network, and there's a number of different things that we did with the tool. Essentially, if you want to find out more information about how the two tools I'm just going to briefly mention today, we've got a poster that Josh Noirel, who's in the red shirt in the audience there, is hopefully going to be standing next to later, so he can get a little bit more information. But essentially, what we're trying to do is we're trying to look at, how many up or down regulated pathways, for example, were in a particular up or down regulated proteins were in a particular metabolic pathway within certain rules, and we wanted to make sure that we were avoiding traversing too many hub currency metabolites, and this was essentially how the network could be reassembled and hopefully use some prior information to hopefully generate the connected components in the pathway. We applied this to nitrogen fixation in this particular cyanobacterium because this is one of the organisms that we were potentially looking for originally for a synthetic biology chassis. And the interesting fact is that during nitrogen metabolism, during nitrogen fixation, hydrogen is produced as a byproduct, and so this was obviously something of interest, and we wanted to find out, at least in the native state of the cell, before we did any modification, how did this actually work. What you want to be able to do, of course, is perhaps have a very, very thorough understanding of the network structure in an organism. And this is actually a reconstruction in a cyanobacterium of a network structure, but it's qualitative, and this took months to do manually. Okay, so this was a particular problem. When we applied the heuristic tool, we were able to generate this particular network here of the nitrogen fixation process, and we're able to look at various up and down regulated pathways, and so the red and blue dots represent the up and down regulated proteins that we're able to look at in the network structure. The open circles are things we didn't find, so it is a sparse network, as I said. What we're able to do as part of this is that we're able to generate this particular network here, which was essentially joining together glycolysis, the pentose phosphate pathway, with nitrogen metabolism. This is nitrogen, this is pentose phosphate pathway side. The interesting thing about joining these two parts of metabolism is that they were joined through this part here. And the interesting thing about this is that we wouldn't have been able to do this without the heuristic model, because we didn't find either of these two black dot proteins here, and the yellow here was below the statistical cutoff for determining it was upregulated or not. And so the model did allow us to join these two particular parts of metabolism together. We subsequently redid the proteome, and we were able to identify and quantify these proteins on a biological set of biological replicate cultures, and indeed showed that this model was actually what we observed from real data. So there are a number of limitations of this model, is that the results are dependent on several factors. The quality of the metabolic reconstruction, so if you take something from KEGG, or you take one of the Palson models, or you take another model, of course, you're going to get different answers depending on what the reconstruction looks like. The quality and coverage of the mass spectrometry data is a problem, because the more sparse it is, the harder it is to actually get anything out of the data, and also the data can be quite noisy, and so that's also a particular problem. And some problems also eliminate from the inconsistencies or limitations of the metabolic network, which result in unestimated weights. And so this is in terms of dealing with how one traverses the metabolic network. And I think one of the biggest problems, in fact, in all of this is that you've got no idea when you actually use, certainly this heuristic model, you've got no idea whether the unobserved proteins that you estimated were up or down. You've got no idea whether, in fact, that's true in reality unless you go out and carry out an experiment. So there's no probability assigned to whether these things are true or not. And so to solve this problem, we decided on a Bayesian approach to actually try and to try to apply this statistical tool to really try and get an understanding about the prior probability of in fact those proteins being up or down regulated from your model predictions. And we were certainly able to demonstrate that it was indeed possible to apply a full Bayesian approach to be able to use this sparse data and to realistically obtain an idea of the reconstruction of the network. I'll skip those things in the interest of time. The further information is available on the poster, but I just wanted to say that what we're able to do is we're able to construct a network that was very, very similar, in fact, to the network we generated from the heuristic model. And again, we're able to have this model traverse these two particular parts of metabolism, energy metabolism, and the metabolism from glycolysis. And so we were very, very relieved that we're able to generate a very, very similar network, which we're able to validate experimentally in a subsequent run. What we're doing next really is to look at the RNA data and to combine the RNA behaviour and the proteomic behaviour to try and get a more systems-wide understanding of what we're actually observing in the cell. As I said before, the correlation between what you observe changing at the RNA level and what you observe changing at the proteomic level is quite poor. They're very, very poorly correlated. Here's a particular example here where you've, I can't remember which way it's around, but you've got proteome changes on one side and RNA changes on the other, and you can see that it really doesn't at all follow the one-to-one line, and this is very, very typical. This isn't our data, this is from the literature, but we've observed this in all of the systems that we've studied as well, so it is a particular problem. If you want more information on what we're doing, there's two posters presented here. This one from Joss describes how the model works in a little bit more detail, and explains a little bit more about what we've been doing with the RNA data. And here is some more proteomic-based stuff from Karen Van Elberg, who's been looking really at trying to develop tools to get wider proteome coverage. The model itself for the Bayesian analysis is available, is written in R, and it's available on the internet for full download, so that you can implement it in your own studies if you so wish. So in conclusion, post-genomic tools such as proteomics can give real systems-level insight into bioprocess generation, bioproducts generation processes. There is now becoming some addressing some of the computational and technical limitations of the omics approaches, particularly proteomics, to give better network reconstruction and understanding, hopefully. And systems biology understanding is providing a springboard for synthetic biology construction, characterisation and exploration. And with that, I'd just like to thank a few people that have really done the work. Joss is our, I guess, captive bioinformatician who's done a lot of the work in developing these tools. Guido Sanguinetti has also been intimately involved. He was in computer science at Sheffield, and now he's joined us in chemical engineering and working on these particular models. Saul Yanao has done all of the bench scale proteomic work, along with Karen Stenzo from Uppsala University. And I'd like to thank the European Union primarily for the funding of this work. Thanks very much.
Before going to the questions, I want a lot of people sitting on the aisles. There is space here. You just can sit, and it's all right. You just pass it here, you sit, and you are much more comfortable. All right. So I suppose there are questions now to Phil. Thank you. Phil gave us the first springboard of going to omic technology and analysis for the synthetic biology. That will follow up in the second two second presentations. On Ping, please.
Can you tell us a little bit more what you have done on that project, or what is planned? I mean, that would be very interesting.
What we've done is there's been some various. We're working mostly in Synacha cystis now, and there's been we've done various characterisations of the behaviour of Synacha cystis in its native state in producing hydrogen. As I said, we've been trying to develop the tools to actually characterise this possibly, and now we've been able to do that. The poster that Karen's presenting out in the poster session really gives some idea about some of the characterisation work. We've got a number of different mutants where there's been several genes knocked out in the particular chassis to look at the impact of the hydrogen work. And also Uppsala University has got a number of posters describing what types of knockouts, what types of plasmids, etc. have been done in Synacha cystis, and all of those are available out there. So, there's... Alfonso, how many of us are there on the biomodular here? There's quite a few, right? I don't know where he's gone, but there's at least half a dozen posters out there which really summarise it all.
Further questions? So I have a more philosophical question. So because what we need is going beyond the systems biology, as you say, is a springboard. But what is then, in your view, what is specific that we need in synthetic biology using your modeling frameworks? Let's say we need specific tools for synthetic biology, for design, computational design of experiments, of systems, parts and devices that you showed before. In what way do you think you could extend your methodologies to help us in developing specific tools for synthetic
I think one of the problems is going to be is a lot of the, you can design some of the things that you want to put in, but we don't necessarily know what the interactions are going to be if you start making large-scale changes to a chassis. And also if you take some of the parts and devices from one chassis and put them in another one, what that actual impact's going to be. And so I think, certainly from my point of view, trying to understand the larger scale changes that happen when you actually make some of these changes are important. And I know there are modelling tools that are coming out now to help to do that. And so I think it's still going to be this iterative process between these new design tools that predict the metabolic changes, predict the network changes, and then hopefully between what we're able to measure and what these new design tools can do, hopefully this can be iterative to try and really improve them so that maybe we don't need to do the experiments, because the experiments are obviously a hassle and they're expensive. And so, you know, it might do away with it if the computational tools at these interactions can be improved, I think.
Well, still, you know, models are just, you know, simplification of realities, not realities. So I think most of us notice that how difficult it is to make a prediction, and it's very easy, relatively easy, and you go to the lab and somehow it doesn't work.
Yeah, exactly.